Good day, this is Professor Will, CBMD PhD. Today's Friday, February 28, 2020. It is 11:45:49 seconds p.m. Eastern Standard Time. And this is going to be a short reading on virulence genes. So let's get started here. Studies of the A-tumefacient's infected process have focused on the roles of Ti plasma genes and tDNA transfer and opine response. Several essential and ancillary chromosomal genes also have been shown to contribute to A-tumefacient's pathogenicity. Although mutations in these genes are often pleiotropic, they generally function to regulate virulence gene expression or immediate attachment to plant cells. At least 3 groups of chromosomal genes have been identified that activate or repress virulence gene expression. As described previously, the paraplasmic sugar binding protein, CHVE, complex with any of a wide variety of monosaccharides induces conformational changes in virulence A, allowing it to interact with phenolic inducers. Interestingly, CHVE mutants are not only severely compromised for tDNA transfer, but also show defects in chemotaxis towards sugars, suggesting that CHVE interacts both with virulence A and with another membrane proteins involved in chemotaxis. CHVE therefore plays a dual role in the physiology of A-tumefacients by promoting chemotaxis toward nutrients and by enhancing the transfer efficiency of opine encoding tDNA to plant cells. A second locus codes for ROS, a transcriptional repressor of certain virulence operands. As described later, the virulence C and virulence D operands contribute to the tDNA processing reaction. Although the promoters for these operands are subject to positive regulation by the virulence A and virulence G transduction system in response to phenolics and sugars, they are also negatively regulated by the ROS repressor. A mutation in ROS leads to constitutive expression of virulence C and virulence D in the complete absence of virulence G protein. ROS binds to a 9BP inverted repeat, the ROS box residing upstream of these promoters. In the absence of plant signals, ROS binding to the virulence C and virulence D promoters prevents the tDNA processing reaction. Whereas in the presence of plant signals, ROS repression is counteracted by the virulence A and virulence G induction system. Interestingly, ROS was recently shown to be a novel prokaryotic zinc finger protein that functions to repress not only the expression of tDNA processing genes in the absence of the suitable plant host, but also the expression of the tDNA oncogenes in the bacterium. A second 2-component regulatory system has been identified that, like the virulence A and virulence G transducer pair, senses environmental signals and mounts a behavioral response to modulating gene expression. All mutations in genes for these proteins result in cells which cannot induce the virulence genes or grow at an acidic pH of 5.5. The molecular basis underlying the effect of the CHVG and CHVI proteins of virulence gene expression is unknown. Binding of the A-tumefacients to plant cells is required for tDNA transfer. Recent evidence indicates there are at least 2 binding events that may act sequentially or in tandem. The first is encoded by chromosomal loci and occurs in the absence of the Ti plasma gene. This binding event directs bacterial binding to many plant cells independently of whether or not the bacterium is competent for exporting tDNA, but a given plant cell is competent for receipt of tDNA. The second binding event is mediated by a pilus that is elaborated by the virulence B gene. Binding via the chromosomally encoded attachment loci is a 2-step process in which bacteria first attach loosely to the plant cell surface, often in a polar fashion. A series of genes termed ATT are required for the binding reaction. The second step involves a transition resulting in the tight binding of the bacteria to plant cells. The CEL genes that mediate this form of binding direct the synthesis of cellulose fibrils that emanate from the bacterial cell surface. Recent studies indicate that binding due to these chromosomal functions occurs at specific sites on the plant cell surface. Binding is saturable, suggestive of a limited number of attachment sites on the plant cell, and binding of virulent strains can also be prevented by attachment of a virulent strain. Although the identity of a plant cell receptor has not been definitively established, a good candidate is a vitronectin-like protein found in detergent extracts of plant cell walls. Attachment proficient A-tumefacients cells bind radioactive vitronectin, whereas attachment deficient cells do not bind this molecule. Intriguingly, human vitronectin and anti-vitronectin antibodies both inhibit the binding of A-tumefacients to plant cells. Efficient attachment of bacteria to plant cells also requires the products of 3 chromosomal loci, CHVA, CHVB, and EXOC and PSCA. All three loci are involved in the synthesis and transport of cyclic B-1,2 glucan molecule. Mutations in these genes are pleiotropic, suggesting that B-1,2 glucan synthesis is important for the overall physiology of A tumefaciens. Periplasmic beta-1,2 glucan plays a role in equalizing the osmotic pressure between the inside and outside of the cell. It has been proposed that loss of this form of glucan may indirectly disrupt virulence by reducing the activity or function of the cell surface proteins. Interestingly, CHV mutants accumulate low levels of virulence B10, one of the proposed components of the T-complex transport system, suggesting that B-1,2 glucan might influence tDNA export across a bacterial envelope by contributing to transporter assembly. One of the early events following attachment of plant cells and activation of virulence gene expression in response to plant signals involves the processing of tDNA into a form which is competent for transfer across the bacterial cell envelope and translocation through the plant plasma membrane, cytosol, and nuclear membrane. The prevailing view, strongly supported by molecular data, is that tDNA is transferred as a single-stranded molecule that is associated with both covalently and non-covalently with virulence proteins. Two proteins identified to date are components of the transfer intermediate, Virulence D2, an endonuclease that participates in the tDNA processing reaction, and Virulence E2, a single-stranded DNA binding protein which is proposed to associate non-covalently along the length of the single-stranded transfer intermediate. Intriguingly, recent studies have provided strong evidence that A. tumefaciens can export the Virulence E2 SSB to plant cells independently of tDNA. More than a decade ago, investigators determined that the tDNA border repeats are cleaved by a strand-specific endonuclease and that the right tDNA border sequence is essential for and determines the direction of DNA transfer from A. tumefaciens to plant cells. The predominant product of this nicking reaction was shown to be a free single-strand tDNA molecule that corresponds to one strand of tDNA. It was noted that these features of the tDNA processing reaction are reminiscent of early processing events involved in the conjugated transfer of plasmid between bacterial cells. In the past 10 years, a large body of evidence has accumulated supporting the notion that DNA processing reactions associated with tDNA transfer and bacteria conjugation are equivalent. Extensive studies have shown that two systems, in particular the tDNA transfer system and the conjugation system of the broad host range plasmid RP4, are highly similar. The substrates for the nicking enzymes of both systems, tDNA border sequences and the RP4 origin transfer ORIT, exhibit a high degree of sequence similarity. Furthermore, the nicking enzymes Virulence D2 and PTI and TRAI of RP4 possess conserved active site motifs that are located within the N-terminal halves of these proteins. Purified forms of both proteins cleave at the nick sites within tDNA borders in the RP4 ORIT respectively. In the presence of Mg2+, purified Virulence D2 will catalyze cleavage of oligonucleotides bearing a tDNA nick site. However, Virulence D1 is essential for nicking when the nick site is present on a supercoiled double-stranded plasmid. Both Virulence D2 and TRAI remain covalently bound to the 5' phosphoryl end of the nicked DNA via conserved tyrosine residues, Tyra29 and Tyra22. Finally, both proteins catalyze a joining activity reminiscent of type I topoisomerase. Virulence D1 was reported to possess a topoisomerase I activity, but recent work suggests instead that Virulence D1 supplies a function analogous to TRAJ of RP4, which is thought to interact with ORIT as a prerequisite for TRAI binding to an ORIT DNA protein complex. The current model describing the tDNA plasmid conjugation processing reaction is that sequence and strand-specific endonucleases initiate processing by cleaving at tDNA borders and ORIT sequences, respectively. This reaction is followed by a strand displacement reaction, which generates a free single-strand transfer intermediate. Concomitantly, the remaining segment of tDNA or plasmid serves as a template for replacement synthesis of a displaced strand. It is important to note that the single-stranded transfer intermediates of the tDNA and RP4 transfer systems remain covalently bound to their cognate endonucleases. Considerable evidence suggests that these protein components play essential roles in delivering the respective transfer intermediates across the bacterial envelope. Autoinducer. An acylhormylserine lactone secreted from bacteria which under conditions of high cell density passively diffuse across the bacterial envelope and activates transcription. Order sequence 25 BP direct imperfect repeats that delineate the boundaries of tDNA. Conjugal pilots. An extracellular filament encoded by a conjugated plasmid involved in establishing contact between plasmid-carrying donor cells and recipient cells. Metabolizable plasmid. Conjugal plasmid that carries an origin of transfer ORIT but lacks genes coding for its own transfer across the bacterial envelope. Transfer intermediate. A nucleoprotein particle composed of a single strand of DNA destined for export and one or more proteins that facilitate DNA delivery to recipient cells. Good day, this is Professor Will, CBMD PhD. Today, Saturday, February 29, 2020. It is 1:11:17 seconds a.m. Eastern Standard Time. And this concludes my brief lecture on encoding genes. Hope you enjoyed listening. Have a great day.